Respected dignitaries, I am Professor Geetha, working as Professor and Head, Department of Botany, Bada University, Coimbatore. My research topic is Biogenic Nanoparticles for Plant Disease Management and Sustainable Agriculture. So morning session we have heard a lot about nanotechnology and applications from Dr. K.S. Subramaniam talk. So now here I want to share with you the basic technology, basic things only, just synthesis, characterization, how these various types of nanoparticles used in agriculture include not only agriculture, human welfare also. I have concentrated first. Then nowadays I am doing some of the work related to plant disease management. Just I want to share with you. So this is the content of my topic, that is nanoparticles, types of nanoparticles, advantages and disadvantages of nanoparticles, methods of synthesis of nanoparticles, then characterization of nanoparticles, activities of nanoparticles in plant disease management and key applications in agriculture, proposed mechanism of nanoparticles to mitigate biotic stress in plants, then type of nanoparticles used in plant disease management, and conclusion. These are my publications. I have done research on nanoparticle synthesis and characterization and application for the past 10 years. Recently, we published one paper with the help along with Dr. Sendil Raja. Here, we made chitosan nanoparticles loaded dimethoxycurcumin, and we applied these nanoparticles against Phytophthora infestans to evaluate the antibacterial activity of potato blight disease. So, what is nanoparticles? So we know very well food security and sustainability are seriously threatened in the world because of the increasing world population. It is estimated that the world population will be increasing up to 10 billion by 2050. Yield of crops are affected due to different biotic stresses, but which they could be effectively managed by application of chemical methods, but these methods leads to toxicity to human as well as environment. As a result, we need some innovative methods. Nanotechnology is one of the innovative science. Approximately 16% global agricultural production lost taking place due to phytopathogens. Fungi only accounting for about 70 to 80% of the yield loss. So, green nanotechnology, it means synthesis of nanoparticles from plant pods, is a eco-friendly technology. It is widely accepted in agriculture. It refers to utilization of nanoscale materials, that is 1 to 100 nanometer, synthesized from living organisms. Nanoparticles, there are three major types. One is, it is classified based on composition they made: organic, inorganic and carbon based. Organic nanoparticles made from lipids, proteins and carbon. They have own advantages and disadvantages. For example, organic nanoparticles are often non-toxic and biodegradable, but they are sensitive to thermal and electromagnetic radiation. Inorganic nanoparticles made from silica, gold, silver, some metal oxide also. They show highly organized atom arrangement and also show more stability in nature. Some special unique properties like antibacterial activity, antifungal activity. Carbon based nanoparticles examples include quantum dot, fluorines, graphene. They show distinct mechanical, chemical and physical properties. So, there are two common strategies for synthesis of nanoparticles. One is from top-down method, another one is called bottom-up method. In top-down method, nanoparticles are synthesized from bulk materials. In bottom-up method, these nanoparticles are synthesized from atoms, small particles. Under top-down method, physical method is coming. In physical methods, we can prepare nanoparticles using sophisticated instruments like ultrasonication, laser ablation, ball milling, sputtering. In bottom-up method, chemical methods as well as biological methods are coming. In chemical methods, we have to use chemical reduction and electrochemical process. Biological methods, we can use plant, fungi, even bacteria mediated synthesis. This method is highly stability and biocompatibility of nanoparticles we can synthesize. So once nanoparticle synthesis in the lab, then we have to go for confirmation of the nanoparticle synthesis. Initially, when we add the plant extract along with the precursor solution, immediately color change will be observed. So not only color change will not confirm the formation of nanoparticles, therefore we have to go for various basic as well as Advanced spectroscopic characterization. First one is UV visible spectrum. If you analyze the nanoparticle solution under UV visible spectroscopy, you can get the SBR band formation, which is not absent in the precursor solution. So it confirms the formation of the nanoparticles. Next is transmission electron microscope. Here you can find out the exact size and morphology of the nanoparticles. XRD analysis, you can find out the size and the crystallinity of the nanoparticles synthesized. Scanning electron microscope will tell the surface morphology of the nanoparticles. Dynamic light scattering will say the size, distribution and dispersity of the nanoparticles. Atomic force microscopy will give the three-dimensional structure of the nanoparticles. FTR spectroscopy will give idea about the functional groups present on the nano solution. So this characterization is very important before applying these nanoparticles for various purposes. So what are the activities of nanoparticles in plant disease management? Nanoparticles have many properties like nematocidal activity, insecticidal activity, fungicidal activity, and activation of plant defense mechanism, bactericidal activity, and antiviral activity. There are three key applications in nanotechnology. Morning sir discussed, case Subramanian sir discussed. They can be used as nanopesticide, nanofertilizer, and nanosensor. Silica and silver are mainly used as they act as insecticide because they will disturb the exoskeletons of the insects. Next, nano fertilizers. Some examples: nano urea, nano DAP, nano potash, nano apatite. Nano urea contain liquid form of nitrogen. Nano DAP contain phosphorus. Nano potash contain potassium. Nano apatite contain both calcium and phosphorus. Then nano sensors, that is, it will with the help of nano sensor we can do precision agriculture or smart agriculture. We can monitor disease and insects, pollutants, soil nutrients, and water contents and temperature. Everything we can control. We can control from the real-time monitoring basis, and also we can assess the crop growth and yield. So what is the proposed mechanism of the nanoparticles to mitigate the biotic stress in plants means nanoparticles act on plants in combination of direct and indirect activity. Direct means they show direct antimicrobial activity. In this, nanoparticles, whenever attaches to the plant cell, they will disturb the cell wall and cell membrane and leads to death of the insects. The indirect means here these nanoparticles once binds with the cell membrane, it will enhance the cell membrane of the cell membrane and cell wall of the plant cell, means it will induce the immunity of the plant. So automatically plant will withstand the particular stress condition. So this is the antibacterial mechanism of the silver nano. For example, I am telling here antibacterial mechanism of the silver nanoparticles. If you apply the silver nanoparticles in the liquid form to the plant, suppose if you give spray, means first it will disturb the cell wall and membrane of the insects. Then second, they will once they enter the inside, they will attach to the ribosomes and make the denature. Third one, they interrupt the ATP production. Fourth one, they disturb the membrane and also forms lot of reactive oxygen species. This reactive oxygen species interfere with the DNA replication. Then finally membrane and denaturation is taking place, and then all the cellular things coming out of the cell, it leads to death of the insects. Then type of the nanoparticles used for plant disease management so far: silver nanoparticles, copper nanoparticles, chitose nanoparticles, zinc nanoparticles. But as silver, copper, zinc, they are somewhat metal based nanoparticles. Sometimes morning we discussed about toxicity problems. Still now we don't have any regulatory frameworks using these nanoparticles large level to field conditions. That's why alternatively we are preferring to use chitosan based nanoparticles, because chitosan is derived from the chitin of the insects. So from this chitin we are processing and we are getting the chitosan. This is biocompatible, biodegradable, eco-friendly manner. That's why comparing to other these 4 nanoparticles, we are using only chitosan nanoparticles for our work. So this is the silver nanoparticle. This is the silver nanoparticles. This is prepared from various from lower plants to higher plants. We can synthesize. This is the basic mechanism of the nanoparticles. For any nanoparticle synthesizing, the following this method only. Suppose if you take plant leaf, from this leaf you have to make the leaf extract. Maybe aqueous extract you can take it. After that you have to mix with the that is precursor solution. It means silver nitrate is the precursor solution. If you mix means automatically some color change will be taking place. It indicates the indicates the formation of the biosynthesis silver nanoparticles. So here why it happens means plant extract contains some phytocompounds. These phytocompounds like phenol, flavonoids, terpenoids, tannins. These phytocompounds acting as capping as well as reducing agents. What is reducing agents? That is, for example, silver nitrate solution I am mixing with plant extract means the silver ion converted into the silver nanoparticles. This conversion is known as a reduction division. This is happened due to the action of the phytochemicals. Next, once nanoparticles are synthesized, they aggregate together and begin to grow. That process is known as nucleation. Then automatically they have to withstand for some time, at least for 3 months, 6 months. It has to stable. So in order to give this stable, these phytochemicals, some of the phytochemicals again encloses these nanoparticles. That process is known as capping. Okay, this is the methodology mechanism of the synthesis of any nanoparticles. So like this we can prepare the copper nanoparticles. We can prepare the zinc oxide nanoparticles. So suppose if you evaluate any plant disease management, for example, one of the studies we applied the biosynthesis zinc oxide nanoparticles to control the mycelial growth of the aspergillus niger of the aqueous extract of the Symbobogon citatus, that is lemongrass extract we have taken. From this we synthesize the nanoparticles. Various concentration we have used. Out of these concentrations, 200 microgram per ml zinc oxide nanoparticles showed less growth of the mycelium. So I already told you, among these silver, zinc and copper, we prefer only chitosan nanoparticles because chitosan is a sugar that comes from the outer skeleton of the shellfish, including crab, lobster and shrimp. Chitosan is derived from chitin through the process called deacetylation. It means which remove the N-acetyl groups and also to create the free amino groups. These amino groups will easily attach with any phytocompound or any phytoextract. That's why we are encapsulating, we are loading the phytocompound or phytoextract onto chitosan nanoparticles. Because of this property only, it will show the adsorption property. So this is a single method of preparation of chitosan nanoparticles. Here ionic gel method is used because first initially we have to get the chitosan solution. It is commercially available. So once we have to get chitosan, we have to dissolve in small volume of the 1% acetic acid, glacial acetic acid to dissolve properly. Then we have to add the cross-linking agent that is called TPP, trisodium polyphosphate. This is the chitosan itself is a negatively that is positively charged molecule. TPP is the negatively charged molecule. So once we mix both the things means automatically cross-linking will be taking place. They attach each other due to electrostatic interactions. As a result, we are getting the chitosan nanoparticles. Once chitosan we obtained, we have to go for thorough washing with the help of water. 2 minutes. Okay, water. Then we have to go for centrifugation. We have to filter it. We have to dry it and store it properly. Whenever we want, we have to dissolve in small volume of DMSO. Then in water, we will get the fine without any precipitation solution. This also same with the as similar to the nanoparticle mechanism only. It is also following. So here the chitosan, now once chitosan nanoparticles I prepared, I want because one chitosan nanoparticles did not show any specific activity. When you attaches any plant extract or plant phytocompound means it act as a carrier. It will go and bring the and deliver the drug to the particular region only, specific region. That's why we are loading the phytocompounds or plant extracts onto chitosan nanoparticles. So conclusion, morning we already discussed, there are a lot of toxicity problems using nanoparticles. That's why we stopped synthesize these metal and metal based nanoparticles. I am concentrating only on chitosan based nanoparticles only. Metal based nanoparticles, suppose if you use before use in large scale means we have to go for more research, and also we have to sanitize the optical application and long term environmental impact also we have to study properly. That's all.
Long live India and France friendship.